So hello everyone, I'm Evangelos. I'm a postdoc at the University of Bath. I did my PhD at Sam Shepard's group, and today I'm going to talk about my latest PhD paper, which was published in May. And let's see what it is about. It's about Campylobacter. So there are some facts that some people may not know about Campylobacter. So it is the leading cause of gastroenteritis in high-income countries. And most of the people know or have heard of E. coli, Salmonella, and Listeria as bacteria that you can get from food, and you can get a nasty disease. It might kill you, and people are scared of them. Not a lot of people have heard of Campylobacter, but it causes 3 times more cases than all of these 3 bugs combined. It is carried in the feces of chickens, pigs, cattle, and wild animals, and around 1 in 7 people suffer from an infection at some point in their life. It's also estimated to be present in the feces of 20% of cattle worldwide. So this slide illustrates the dynamics from which we have Campylobacter being transmitted from one host to another, from the environment to different wild animals, to livestock animals, and eventually humans, if they get sick. And even though this is called the Chicken Journal Club, today it's going to be about cattle. Chicken is the usual suspect, and most of Campylobacter cases have been linked to chickens. And a recent study has shown that if you go to the supermarket and you take a sample, the rates of Campylobacter are really high, actually very scary. Going back into the cow, or the cattle in general, if you think about numbers, it's a bit shocking. So cattle, they take around 50% of Earth's land area, and they consume around 30% of all fresh water. They're also responsible for around 50% of the global greenhouse gas emissions. So the impact of agriculture of ruminants is very high on the environment. But in terms of biodiversity, we have a lot of cattle in the world. So cattle, they constitute more biomass than all wild mammals combined, which is also very shocking. And then you think about bacteria. Okay, you have a cow, it's like an incubator, and you have nice steaks, you have the milk, you have the global trading networks. So for bacteria that live within these animals, this is an opportunity to proliferate and spread globally through these trade networks. And this carries a significant risk, as pathogenic bacteria can potentially spread to humans and cause disease and outbreaks. Right. So, as I said, Campylobacter bacteria, you can isolate them from different livestock animals, different wild animals, but also from the environment. So if you get all these, you go around in farms and you sample Campylobacter, then you sequence the genome, and you start comparing those genomes, you end up with a population structure. It looks like this. So every color represents a different source. So if yellow is the chicken, and the blue are ruminants, and black is wild birds, you get sequence clusters that are associated with different sources. So if you get sick and you go to the hospital and you get sampled and it's Campylobacter, you sequence the genome, then you can try and attribute the source of infection. So in this graph here, you see that for those isolates, the probability of them being isolated from chickens is almost 100%. Here you have some isolates where you have a high probability that they came from ruminants, cows. And here in the middle, you don't really have a good source signal. And this means that you can't really attribute the source in those isolates. And this is what we call some clusters that you can isolate, that you can find in multiple sources, like this 45 here or this 21. So these is what we call the host generalist clusters, and they're tricky to attribute to a single source. Happening. Am I still there?
Yep.
Okay, cool. Right. So what is the contribution of cattle-associated Campylobacter to human disease? So, as I said, most of them come from chickens, but we do have a lot of Campylobacter attributing to cows. So there have been several attribution studies which pinpoint the chicken as the dominant source of Campylobacter. But in a recent study with French collaborators and Sam's group, they have examined the source of a large number of isolates by looking at the allelic variation across the whole genome. So, looking at the graph here, is it working?
Yep, it's working. Yep, thanks.
We see that most of the British clinical isolates, they come from chicken, followed by cattle. But when it comes to French isolates, we have an actual equal distribution. So a lot of the isolates, they do come from cows. So you might have variation within different countries, depending on what they eat and the culture. Also, sometimes you have some weird sources of infection, like if you go out cycling, you go past the field covered with sheep poo, and you have your water bottle, you might get some poo on the bottle, and then you drink, and then you yeah. You have some lag when changing slides. Has it changed for everyone? Because it hasn't for me.
No.
Any questions while we wait for my screen to respond?
There we go. It's changed now.
Okay, perfect. Right. So Sam's group has been interested in cattle adaptation and the genes associated with this. And this is more or less when my PhD project started. So in this particular study, they used, I think it was the first GWAS to be used in bacteria, and they identified the 7-gene region with the host association signal. So 3 of those genes encoded for vitamin B5 synthesis, which allows better growth of cattle isolates in cattle environment. And they did this in this so-called host generalist clusters, where you have isolates from chickens and isolates from cows. So 7 years later, this paper pops up, and we looked at cattle-associated, specific cattle-associated lineages. So 2 of the authors, I think, are in the panel. 2 authors. Ben is here, and Keith is here as well. That paper came out in the beginning of May, which was unfortunately during the COVID high rate in the UK and all over the world. And it got a lot of media attention, British media, but also media from all over the world. Lots of tweets on Twitter and lots of articles online. Right. So we don't really know a lot about how zoonotic bacteria evolved to exploit the change in host niche phase. And what do I mean by that? We have a cow. So looking at the graph, this says 8,000 years before Christ. So this is around the time of domestication of cattle. And since then, here you have an estimate of the numbers of domestic cattle in the world. And in a very recent, from an evolutionary point of view, time, we have an expansion or an explosion of that host niche space. So if you think of bacteria, and let's take Campylobacter as an example, the most abundant human bacterial pathogen found in cattle is Campylobacter. So these organisms are present in the feces of around 20% of cows, and at the concentration of 3 times 10 to the 4 colony-forming units per gram. So to make that short, having around 1.5 billion cattle on Earth, and each of them produces around 30 kilos of manure daily, there are around 3 times 10 to the 17 Campylobacter which are excreted to the environment every day. So the magnitude of this in terms of direct environmental contamination and the spillover into the human food chain is big. And while a lot of research is focused on the epidemiology of strains in human infection, we don't really know a lot about how the intensive farming may establish new transmission cycles and influence the evolution of what we call livestock adapted strains, like the one that we're going to talk about very soon. So if you attended the meeting about pubMLST last week, you should be familiar with the very cool new interface. I use pubMLST quite a lot. I do enjoy the new interface. I find it very nice. For people that don't really know what that is, it's a collection of open access curated databases that integrate population sequence data with phenotype information for a lot of different microbial species. I don't know how many, around 100. And one of them is Campylobacter. So from the last time I checked, there are around 100,000 Campylobacter jejuni and coli isolates, and around 50,000 genomes associated with those. And I went to the pubMLST and I have the look. So there are around 5,000 MLST profiles of isolates which are coming from ruminants or collected from ruminants or ruminant-associated products. And around 1,000 clinical isolates are associated with the 2 major cattle Campylobacter lineages. Right, so going back to the paper now, what we did was pretty simple. We went in one of the database. We got all whole genome sequence isolates we could from cattle and from chicken and some clinical ones, and we made a tree. So this is how the population structure of Campylobacter jejuni looks like. And with blue, you have the isolates, Campylobacter isolates from cows, cattle, and with yellow, the ones from chickens, and the white, clinical and other isolates. So pretty quickly, you notice that there is this cluster here called ST61, which is emerging from another cluster, big one cluster here, ST21. Also, you notice some other sequence clusters on the tree that are yellow, which are chicken-associated clusters, and also this, another cattle-associated cluster, ST42. Right, so we wanted to date this emergence. We wanted to see when it actually happened. So between the 1820 and 1975, the number of cattle increased around 8-fold across the world. So we have new farming techniques, we have improved livestock breeding, we have higher stock densities, and this led to amplified food production. But also we have this host population explosion of that niche, and changed the cattle niche, and that has probably favored strains that are adapted, which is what we call the specialist lineages. So our analysis showed that the emergence of this lineage occurred in around 1860, whereas, move the faces here, yeah. So the emergence happened around here, whereas most of the diversification of the sublineages of this clone happened later, around 1925 to 1965, and this coincides with the intensification of cattle farming throughout the 20th century. Our Bayesian skyline model, so an extra analysis that looked at the effective population size, looked that there was a rapid clonal, showed that there was a rapid clonal expansion of that lineage, which is also consistent with the rapid evolution of cattle specialism, which coincided with the industrial agricultural revolution and the intensification of the animal production. This graph just shows the grazing land that is used over time, and it just follows the trend, the number of cattle in the world. It's just such a short period of time, you have this massive explosion there, which is also pretty shocking. Right, so we have a process of host specialization here, but we're also very lucky to have these intermediate isolates. So you have the background population and then the emergence of this specialist lineage, and the existence of these two enabled us to see if that happened gradually. So we had a look at the ST61-like alleles, as we call them, and we see a gradual shifting from a 21 allele to 61 allele. And if you go further and you look at the nucleotide identity, so the distribution, you see that we have the ST21s here, ST61s here, and the intermediates kind of in the middle. So that shows that this specialization happened gradually. It hadn't happened gradually, and these were what we call hybrids. This would look more like a binomial distribution. Half of them would be ST21 and the other half ST61. Some basics about horizontal gene transfer before we go further. So horizontal gene transfer occurs by different means. As you all know, we have transformation, where the recipient bacterium takes up the extracellular DNA from a donor. We have conjugation, where temporary direct contact leads to the exchange of plasmid DNA. And then you have transduction, where the donor cell DNA is transmitted by a So horizontal gene transfer leads to either replacement by homologous DNA with sequence from another lineage, what we call homologous recombination, or insertion or deletion non-homologous genetic material via non-homologous recombination. And like mutation, horizontal gene transfer can disrupt translation, can cause frame shifts, which can lead to loss of function, but it also has the potential to introduce or duplicate genes or alleles, conferring novel function. So, to summarize what we did in the paper in just one slide, this is probably the less fancy but also most important slide of the talk. We wanted to identify genomic changes associated with host adaptation to cattle. So what we did is we compared the two populations, the ST61 lineage and the ST21 lineage, background lineage from which it emerged from. So we said, okay, what are the genes present in 61, absent in 21, and the other way around? And we know that Campylobacter recombines a lot. So we wanted to see how many genes are recombining in 61, but not in 21. And then we end up with a pool of candidate genes that are possibly associated with adaptation to cattle. And then we go and detected homoplates. So we wanted to see how many of those genes, our candidate genes, are present or absent in other cattle-associated lineages, like ST42, for example. Why we did that, and what is homoplasia? So it can be very challenging to differentiate adaptive genomic changes that provide an advantage in the host from changes resulting from genetic bottleneck and genetic drift. So among the most compelling evidence for genome adaptation occurs when divergent lineages like ST61 and ST42, which are placed differently on the tree. They probably have different evolutionary history. So when these divergent lineages independently acquire convergent genetic changes, which are not present in the common ancestor, these changes should be absent in ST21. This is what we call homoplasis. So if these genes are present, these two different, for example, lineages here, they should be, that should be evidence that they have to do something with the host or the environment. And this graph shows that. So it might look a bit complicated, but it's not. So this is the relative abundance of genes. So this shows gene present and absent. And that's just the difference. So you see every circle here is a gene. You have a lot of circles here in 0. That means that that gene is 100% present in ST61, 100% present in ST21. So if you take the difference, that's 0. So that gene is placed here. So if a gene is present 100% in ST21, 0% in ST61, you take the difference, that's 100%. So it should be placed here. So we wanted to take those genes at the top and at the bottom. So those genes up here, black ones, should be 100% present or more than 95% present in 61 and more than 95% absent in 21. And the ones that are red, those are the homoplacious ones. So those are the ones that are found in other cattle-associated genes and the ones that we want to focus further on in function. This bit here, that shows the recombination analysis. So every bar here is a gene involved in recombination, and where you have these little triangles, those are homoplacious alleles involved in recombination. So if you ignore this bit, which shows just a preliminary automated analysis that identifies homoplaces, we looked at those, we got those homoplacious genes, and we looked across the tree to see if they are present or absent in cattle or chicken isolates of various clonal complexes. And we saw that they are, even with small differences, higher in, more prevalent in cattle-associated strains. And this is also another slide that really illustrates what we want to show. So these are single gene trees of the candidate or the homoplacious genes that we identified. So it pretty clearly shows that if you make single gene tree of that homoplacious gene, you lose the population structure of the tree. So the ST61 and the ST42, they don't really cluster separately anymore. They cluster together. You go to signal. So you have these 4 alleles. So these are recombinant genes. And then you have accessory genes that are present in 61, 42, and absent in the chicken associated genes. Right, gene loss. We talked about, we saw genes that are gained in some lineages. But what about genes that were lost? So we know that when you gain genes, that might be give you an advantage to adapt into a new environment. That can also happen by losing a bunch of genes. And that has been shown in Campylobacter in that paper. And that's for a sequence type 403. But it's also been demonstrated for other bacteria in the literature, I think Salmonella as well. This is not really found here. So we saw that there was a whole gene block that got lost in 61. And that gene block was also absent from ST42, the other cattle-associated genes. And it was present in the background population, ST21, and also in all the other chicken-associated lineages. So that gene block was associated with flagella glycosylation, and specifically for the genaminic acid biosynthesis. Basically, sticking sugars on the flagella of the bacterium. So we identified our homoplagious genomic signatures, and this provides broad information about adaptive evolution, but focusing on specific groups of genes, like the loss of this glycosylation gene block. This provides information about the functional basis of phenotypic adaptation. So it gives us a chance to link what we see in the genotype with what we can see in the phenotype. So we looked at the literature and we saw that post-translational modifications of this flagellin, so the sugars on the flagella, are known to be important in autoagglutination and hydrophobicity, and this is associated with virulence in different gram-negative bacteria and in Campylobacter. Also, viable information has been strongly linked with flagella properties and function. So we wanted to investigate this because we had the isolates. So I got trained up to Sheffield, and together with Professor Dave Kelly and Dr. Aidan Taylor, we did a bunch of phenotype assays. So Aidan was really skillful, talented postdoc. He did allelic exchange mutagenesis. So he deleted that gene block for 2 chicken specialists that had the block. And then he did some initial analysis, and we saw a massive phenotype difference with having or not having that gene block. So we actually got the strains, so all the 61s and 42s, and then some chicken specialist strains that we have, and we saw that there was a significant difference when it comes to cell hydrophobicity, autoagglutination, viral information, and there was a trend difference in autoagglutination, which is a difficult assay to perform because the time point where they agglutinate may vary between different strains. So you might lose, it needs to be a specific spot. I had to do that a lot of times. It didn't work perfectly, but there was a trend in that as well. So the absence of genes in this gene block and the changes that we see in the cell surface charts of the flagellum may enable Campylobacter C.jejuni to attach better to the intestinal epithelium, as changes in the hydrophobicity in the flagella are believed to enable bacterial attachment to either hydrophobic or hydrophilic surfaces. And furthermore, surface structures such as the flagella are also known to stimulate host innate and adaptive immunity. So if you lose the gene block and then the associated phenotypic changes, that may decrease the recognition of those strains by the host immune response. So this is a very good basis to further continue the research and do in vivo experiments to further confirm function, which we do. Right, so going back to the other homoplagious genes and the ones that are were identified with their combination analysis. So there is increasing evidence that allelic variation in homologous sequence, which I'm talking about individual SNPs in genes that are present in both chicken and cattle isolates. So if you have individual SNPs, these can also influence bacterial fitness in the new host species. So here you have some examples of genes that we can make assumptions from the function. You have the vitamin B1 biosynthesis genes, and then you have some molybdenum metabolism associated gene, MOB-A, and also some fibronectin immunogen binding protein. So cattle and poultry, they are both animals, but they are very different in terms of anatomy, physiology, and metabolism. So consistent with this, the variation that we can see in genes is involved in diverse functions. So we have differences in diet in cattle. So vitamin B1 supplements are commonly given in poultry. and also at high doses, which you can also detect in the feces later on. Whereas in cows, you don't really have this supplement. Vitamin B1 is produced naturally, it's naturally synthesized in the rumen, and it's absorbed mostly in the small intestine. So if you take a sample of feces from the large intestine, you'll find really, really low doses. So that can potentially mean that Campylobacter in the large intestine of cattle has specific thiamine synthesis requirements. And that's why cattle associated strains, they have this allelic variation, the same allelic variation in those genes. And similarly, MOBA. So MOBA is a gene that encodes for a molybdopterin guanine biosynthesis protein. So that may allows Campylobacter to use different electron donors. It doesn't matter. It might allow Campylobacter to balance the rate of biosynthesis of molybdenum, which has to do with the supply of potentially to cattle toxic molybdenum. So it has been linked that ruminants that are in pasture and grazing over land, they are prone to molybdenum toxicity. It's quite common in ruminants, so that might have to do with that. But again, these are all speculations about differences in anatomy and physiology, but we have a basis to do further experiments to confirm function. And just to do summary. So in my PhD and in that particular project, we got some better understanding of the ecology and evolution of Campylobacter, and we did that by doing simple things. So in the quantitative analysis, we looked at the accessory genome variation, presence, absence of genes, found that block that got lost, looked at the function, looked at the average nucleotide identity, we looked at the allelic variation, we looked at recombination. Campylobacter likes to recombine a lot. Homoplasy is a strong tool to find host adaptation. And then we moved a step further from the basic genomic analysis. We actually want to zoom in the gene function, and we want to test that in the lab in vitro, which we did recently. I think that was all. Just to summarize that none of this would have been possible without all of these people that have been involved in various steps of the analysis. And I definitely wouldn't be able to do all of this alone. Science is always collaboration and working together as a team. Many thanks to the separate lab team for all support. Any questions?